Welcome back and Happy New Year. We are so delighted to see so many familiar faces. Amitava, Caroline, Deepak, Eva, Marv, we see you out there and thank you for joining us. Today's webinar brings DILIsym Services expert modelers Diane Longo, Grant Generaux, and Scott Q. Siler to describe the development and use of IPF-Sym and NAFLDsym to support the development of effective treatments for IPF and NASH. A few housekeeping notes. We take your privacy rights seriously, and by attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions to the presenters via the question panels on your dashboard at any time. A Q&A session with all speakers will follow the last presentation. Quickly, while people join, I'm going to ask a quick poll question. Which sector describes you best? We'll give you a few seconds to go ahead and answer that. Just 2 more seconds here and let's close the poll. All right, so it looks like we've got a great deal of commercial and industry guests with us today. Again, thank you for joining us. Now let's get right to the powerful science. It is my pleasure to introduce the Chief Scientific Officer of DILIsym Services, Scott Siler. Come on in, Scott.
Thank you, Arlene. And thank you everyone for joining our webinar today. We're really excited to share with you some of the progress and the great work we've been doing in our quantitative systems pharmacology modeling of fibrosis in various tissues. My co-presenters will be Grant Generaux and Diane Longo. Before we really get rolling there, we obviously have to issue our disclaimer that generally summarized says more or less that we are not issuing medical advice. And then just to put a face with the name, as Arlene pointed out, I'm Scott Siler, and Grant Generaux and Diane Longo will also be participating in the presentation today. And of course, we stand on the shoulders of those who come before us and those with whom we work. A fantastic team within our division of Simulations Plus, the DILIsym Services team. And it's the contributions of basically everyone that more or less will be presenting today. So our agenda for the meeting is we'll give you just a brief introduction to our division. We know that most of the folks who participate in these webinars are really keen on kind of the classic offerings of Simulations Plus, and we're still a relatively new addition to the family, so to speak. So just a brief overview there. And then speaking of overviews, transitioning to just an overview of fibrosis and kind of how it's applied correctly and kind of dysfunctionally. And then moving into 2 specific platforms of ours, NAFLDsym and IPF-Sym. And then we'll open the floor for some questions and answers, which should be mediated electronically as opposed to verbally. Okay, so as I pointed out, we are part of the Simulations Plus family. I think 3.5 years now, and it's our family, so to speak, continues to grow as we've had several companies join the family over the years. And really, what it's enabled us to do is to put together a really nice, comprehensive offering across all the different types of modeling and simulation to be able to support pharmaceutical drug development, be it efficacy, be it drug design, be it safety, what have you. And lots of fantastic people behind the science, as well as relationships across industry and academia, all of the above. We're really quite excited about all of that. And one of the things, another that kind of gathering together all these capabilities has enabled us to do as well, is to be able to apply our approaches, our software, our services throughout the entire drug development process, be it kind of the earliest discovery, kind of first in human, to even post-approval. We have various modeling techniques, modeling approaches, et cetera, that can help support anywhere the drug development pipeline you may be. So I wanted to make sure everybody was clear about all of that. And then now kind of to zero in a bit on the DILIsym Services division. You know, we're on board with the vision of Simulations Plus, you know, that really features innovation, respect, integrity, and commitment. And kind of our slice of the pie, so to speak, is what we call quantitative systems pharmacology or toxicology, or QSP, QST type modeling. So really what that is mechanistic mathematical modeling that allows kind of as I said, a bit of a different sort of modeling insight to be derived as opposed to kind of the PBPK pharmacometric sort of offerings that we have in our other areas. Some of the platforms that our division offers are listed here. So our kind of very first and most mature is the drug-induced liver injury, or DILIsym. We've also then kind of staying in the realm of QST, we're currently developing something comparable for kidneys. So that's RENAsym, acute kidney toxicity due to drugs. And then, you know, we've had several offerings as well. Sometimes I describe our development process as branches from a tree. We establish a core competency and then develop from there. So once we had a core competency in the liver, it was clear that we should move into NAFLD and NASH. And so hence you'll hear more about that later today, but that was kind of the genesis of NAFLDsym. Once we established core competency in fibrosis, we moved to the lungs, hence IPFsym. RADAsym, or acute radiation exposure, affects a multitude of tissues, including the liver, the lungs, and the kidney, and so it kind of builds from there. Another really important point here, in addition to the fact that, you know, we have these models available for licensing as well as services projects, is that it's something that is happening with increased frequency. Is you know, another core competency of ours is the ability to develop QSP models, QST models. And so we've had a number of pharma partners come to us and you know, establish a brand new kind of custom built QSP model. And so this QSP model development and consulting projects is, I would say, something that is really kind of a growth area for us. And I just wanted to make sure that the community is aware of that. The modeling community has, in my experience, a kind of a limited amount of tools in our tool belt. And the more tools you have, the more capable you are of doing your job. And so if this is something that interests you, feel free to reach out to us and we'll try to support you. So I have had the pleasure of being in the QSP, QST world since 1999, so that many years ago. And one of the things I found over this time is that everybody seems to define it kind of uniquely. And so it also helps then if we establish, you know, kind of what we're referring to, DILIsym Services division, when we're describing QSP and QST. So I'll just take a second to kind of provide such an overview. Hopefully it's review for some, for others it may be something that's relatively new. But really it's, I more or less mentioned the fundamentals of it earlier, which is it's a mechanistic mathematical model of the underlying biochemistry of a given disease or physiologic state. In this case, actually, we're focused on disease states. So we're describing the biochemistry and the physiology of the pathophysiology of the disease. So what happens, what's the dysregulation of the liver in NAFLD and NASH? What's the dysregulation in the lungs in idiopathic pulmonary fibrosis? So we'll go through and we'll pull out from the literature, what are the key components? And then obviously, you know, as part of the modeling, applying the engineering principles to make sure that we're tracking the quantitation, the movement, the dynamics, and put all that together to then build patients, simulated patients that represent the disease state or states, depending on how much interpatient variability there may be. And then, of course, the next obvious step is to simulate treatment, which then requires us, of course, to simulate the exposure of the drug. And that's obviously one of the places where PBPK modeling really is quite helpful, and that it allows us to predict the exposure of the drug at the site of the target, which is often in the diseases we're talking about here, intracellular. whether it's the hepatocyte or in the lungs. And then, of course, the interaction with the actual drug target or the pharmacodynamics. And that's another important point, is it actually extends beyond that, really. It's not just pharmacodynamics and the direct interaction with the target, but also kind of the mechanisms of action. What are some of the downstream responses as a consequence of interacting with transporter X or enzyme Y? And so we take all that information together, and in that Venn diagram, kind of that center point is the prediction of efficacy that is derived then from assembling all these pieces in the way that we've described here. So that's the sort of approach that we use in constructing and utilizing our QSP, QST models. So now I'll transition to providing just a brief overview of fibrosis. Again, I presume for many of you this is familiar. For others, it might be an opportunity to learn. So just be a really kind of speak to it very briefly. But really, fibrosis or collagen synthesis often is part of a process that is essential and useful when done properly, so to speak. And it's part of the wound healing response. And there are a few, kind of the classic, obviously, is you prick your finger and, then the wound heals and everything resolves. Well, it turns out that there is kind of a multi-step process that this diagram represents a wound that may occur in the intestines. But it's a nice diagrammatic illustration nonetheless of kind of the multi-step process. So you have the cell loss, be it mechanical or otherwise, and upon that cell death, it elicits inflammation to varying degrees. The inflammation begets fibrogenesis, kind of in an attempt to then, if you will, patch the hole, at least temporarily. And then, depending on the cell type, cellular proliferation may ramp up to then kind of take over and fill that gap more physiologically, if you will, rather than leaving the collagen there to kind of serve as a perpetual Band-Aid. Now, there are certain tissues where that proliferative capacity is not available, and there are others where it's dysregulated, or if the cell loss is not temporary, if it's ongoing, there are a number of ways in which dysregulation can lead to then what we would call fibrosis and kind of, an incorrect application, if you will, of the wound healing response. Before we move into that fibrosis realm, I want to provide an example of an actual, an area where I think with which we're all familiar. That's a good example of the wound healing response, and this is the response following a myocardial infarction. So this diagram shows, there's a clot in an artery that leads to a specific section of the heart, incurring ischemia. So limited blood flow, limited oxygen availability, hence kind of making the cells, the cardiomyocytes, prone towards necrosis. There's also an area kind of emanating from that hotspot, if you will, that is an area at risk that may be undergoing a limited amount of access to oxygen, et cetera, hence ischemia. And so as we consider then the response in this sort of situation, this diagram in the upper left kind of illustrates those regions as well. So again, in that ischemic region, in kind of the early stage, there is the necrotic death of the cardiomyocytes. And then in the shoulder, the kind of distal region, there's apoptosis that tends to occur to the cardiomyocytes because of the reperfusion allows then the inflammatory response to come in and provide the potential apoptotic mediators to cells that are potentially bioenergetically compromised. That tends to be a recipe for apoptosis. And then, of course, there's the wound healing response. And so often we describe that in this realm as a scar. And so there's the replacement fibrosis. And oftentimes that cell loss, the cardiomyocyte loss that's been replaced by fibrosis, comes as well with an alteration in cardiac function. This diagram from Wu et al. Shows, for instance, how the ejection fraction of the left ventricle is reduced depending on the size of the infarction. So one of the things that we've done at DILIsym Services is we've built a QSP model, a relatively small one that captures each of these stages of a myocardial infarction. So we can simulate the, the injury to varying degrees in the cardiomyocytes. And then that, of course, begets the inflammation, which begets the fibrosis and leads then to, hopefully, recovery, but potentially with altered cardiac function. So I'll just give you a snippet of it. One of the things that, there, in some ways, there's a ton of available data with which to compare. In other areas, there's limited, just as is often the case in QSP modeling. And you can see in this area, with the simulation results on the bottom and kind of a classic histology sort of clinical data on the top, you can see that initially the fibroblasts, based on the histology score, the number of fibroblasts increases substantially within the first couple of weeks, followed by the collagen response. And you can see then that our simulation of a relatively modestly sized 15% myocardial infarction gives rise to the same sort of timing of dynamics, which is really quite useful, illustrating that we've captured at least the kind of the sequential operation of the inflammation and the fibrosis aspects. And then the quantitation, the amount of the collagen is shown here in a simulated population, one of our SimPops. And so again, a relatively limited number of patients in the Margianowski study, show in the clinical patients in black and the simulated patients in red with varying degrees of the myocardial infarction. And you can see that we're capably predicting that the post-MI collagen response, quantitatively, we're capably predicting it to be in line with what was observed in the clinical patients. So again, this is a good example of, when the wound healing process goes well, that it all goes according to plan. And it's now a model that enables us to explore what happens when it goes awry, as well as treatments, either to help accelerate the wound healing response and improve, hopefully preserve cardiac function, and fibrosis, et cetera. So it's a nice new platform for us, one that we're introducing here and making available to the community potentially as well. So now let's switch tracks back to fibrosis. And I think this is my last slide before I hand it over to Grant here shortly. But again, the dysregulation in this wound healing response can lead to fibrosis. And, so visually you can see then this original illustration, there was just a modicum of fibrosis and collagen extracellular matrix components. But, with increased cellular proliferation, myofibroblast proliferation, or persistent cell loss, the fibrosis is definitely present. Now, bringing this back to NAFLD and NASH and IPF, two of the topics of the day, you can see then that, in fact, a common theme in both of these pathophysiologic states is the ongoing loss of kind of the nascent cell type, not the nascent, the original cell type. In this, obviously for NAFLD and NASH, it's the hepatocytes, and in the lung, it's the alveolar epithelial cells. So the persistent and ongoing death of those cells leads to the persistent activation of the immune system, persistent activation of the fibrogenic response, and fibrosis in the liver, fibrosis in the lungs can manifest. Okay, so that wraps up my section. I'll kind of serve as your moderator here going forward. So right now I'll bring Arlene back to introduce our next poll as we transition.
Great, thank you, Scott. That was a good overview. A quick question for the audience. Is your organization studying or developing treatments for fibrosis? We'll give you a few seconds here to answer. All right, let's go ahead and close the poll now. It looks like you guys are in the right spot to learn more from what Grant is going to bring to us on NAFLDsym. Grant, go ahead and take it away.
Hi, thank you, Arlene, and thank you, Scott. So this next section, we want to give you a fairly quick overview of NAFLDsym, which is our platform, QSP platform for helping investigators and organizations find efficacious medications for NAFLD NASH. So Scott's already talked about this briefly, but just as a reminder that NAFLD is a progressive disease of the liver. It's a disease that starts off through the elevated accumulation of lipids and progresses through an inflammatory state and into an accumulation of ECM. And one of the reasons why industry in general and DILIsym Services and Simulations Plus is interested in NAFLD and has developed NAFLDsym is because it's really a disease of public health concern. It's got sort of a growing worldwide incidence across both the developing and developed world. And it's also a disease for which there are really few treatment options. So that really leads to, really substantial opportunities to make an impact through, aiding investigators and looking at QSP models. So NAFLDsym, again, is a platform QSP model of NAFLDsym NASH. As you can see here, it includes several submodels that address the different aspects of pathophysiology from steatosis through fibrosis. And as Scott has talked about, it has a diverse number of SimPops or simulated patients. So, similar to what Scott talked about, these platforms, all of them, they give the investigators and organizations the ability to combine exposure, pharmacodynamics through the incorporation of their targets, as well as the underlying pathophysiology to understand how treatments will affect the efficacy. Both NAFLDsym and IPFsym are really flexible frameworks that allow us to incorporate new targets, allow us to explore mechanistic hypotheses, and also optimize clinical trial protocols. And so for NAFLD in particular, since it is such a complex disease with lots of different determinants of progression or factors that influence progression, it also gives the ability to look at combinations of different treatments and evaluate their ability to affect outcomes. And to this end, we've actually evaluated greater than 20 different targets and or compounds across, working with several different companies, a few of which are shown there at the bottom. So if we then look at just an overview really quickly of the model itself, of the platform, as it's shown here, it's sort of divided into three different submodels. We have an upper left-hand corner, model that deals with lipid dynamics and steatosis, that accumulation of lipids in the liver. That eventually drives apoptosis and drives a low-level inflammation, which is sort of captured in these blue, sort of items in the upper right-hand corner. And that inflammatory response then drives mediators, which produce extracellular matrix at an increased rate. So it affects the overall balance of ECM. And that submodel that basically tracks the ECM dynamics is in the lower right-hand picture here. As Scott alluded to, we start off, sort of mapping how we understand the physiology and the biochemistry to understand, how these different components that drive pathophysiology relate from a qualitative perspective. And then we layer onto that and leverage clinical data, and in some cases preclinical data, in order to then, understand and put together the quantitative relationships to these different factors and components that make up the abnormal pathophysiology. And a key aspect of doing that is again, the simulated patients, so we refer to as SimPops, and these are a way for us in silico through the modeling to account for the inherent pathophysiologic and clinical heterogeneity, which is seen with the disease and with the clinical outputs. So putting all these things together, it's something that allows us to really do simulated clinical trials and have the ability to predict responses to treatment. So if we look at a little bit deeper into the concept of SimPops, if for any individual simulated patient within a SimPops, each one of them has sort of a unique set of different parameters which are related to different aspects of the pathophysiology as well as normal physiology. And so, for instance, in the list here on the right-hand side, you can see different variables that are related to aspects of the pathophysiology which are important and for which there's known heterogeneity that leads to the way that the disease progresses and expresses itself. So we can see things here like, the lipogenesis, the body weight, the regeneration rate of hepatocytes, the activation of stellate cells, synthesis rate of collagen, etc. So what we do basically is we look at the range of different values that are related to these variables, and we basically we'll do the combinatorics and basically sample from a distribution of these different ranges in order to get a diverse set of different simulated patients. So you can see here now we have our patients with all kinds of different colors to represent these different combinations of these varied parameters. And so, basically what you'd end up with then is patients which, through these different combinatorics, represent, the spectrum of the pathophysiology and the way in which it manifests itself through the progression from steatosis all the way through fibrosis. So one of the things we do after we come up with these patients is we're interested in running the simulations and making sure that the actual outputs from the simulations match with a lot of the data that's measured clinically. So you can see some examples here in the lower left-hand corner where these are not factors that are calibrated on, but they sort of emerge from the simulations. Things like the relationship between fibrosis stage and the liver stiffness, or the ALT level as a representative of lipotoxicity, and the liver fat burden. These are things that we make sure are consistent with what is observed clinically across a range of different outputs and also different clinical studies. And then lastly, the last thing we do is we apply weight loss, which is really the main sort of standard of care in terms of helping to modify the progression of NAFLD-NASH. We apply that as an intervention in order to validate that both our model and our SimPops respond correctly to that intervention. So with that in sort of background in terms of the SimPops in place, we want to go through just a few examples of the different outputs that we looked at across the different submodels. So if we start with steatosis and sort of the steatosis submodel, these are a couple examples of the type of outputs that we look at that really capture the relationships that are seen in NAFLD patients. So for instance, in the upper right-hand corner, we basically have a couple studies from Lambert 2014 and Smith 2019 that establish this relationship between, showing that patients that have extensive steatosis also tend to have elevated levels of de novo lipogenesis. And then within our SimPops, we can see that we're capturing that relationship between those two factors and the range, very well. And then if we continue and we think, another step downstream, we have the relationship between liver fat and ALT representing lipotoxicity on the bottom right here. So again, we're capturing that relationship nicely. Then if we look at the immune system and think about, after the extracellular vesicles, which are downstream from apoptosis of hepatocytes, have started to drive inflammatory response and have the macrophages, for instance, release different mediators, then we're interested to see if we're capturing appropriately those mediator levels. And so these are two examples of really key mediators in the inflammatory response and the fibrosis. In the upper right-hand corner, we have the relationship between ALT as a proxy for lipotoxicity and its relationship with TGF-beta, which drives the actual activation of stellate cells in the liver. So again, the relationship looks quite nice. And then lastly, the relationship between fibrosis stage across different disease severities and the release of PDGF as a mediator, which drives the proliferation of activated stellate cells. And then lastly, after that inflammatory response releases mediators, then it starts to change that dynamics of the synthesis of ECM and collagen. And so we have a couple examples here of the relationships that the model shows and the SimPops show with respect to severity as measured by fibrosis stage and the actual collagen synthesis rates, in this case of collagen 1. So you can see here that with increasing fibrosis stage, you're increasing the synthesis of collagen, which is what we expect and what we understand from the disease progression. And then lastly, we can look not only at the synthesis rate, but the actual absolute density or amount of the collagen in the liver and how that relates to fibrosis stage. And so here you can see, for instance, going from healthy individuals through fibrosis stage 3 in the bottom right-hand graph, you have sort of a subtle increase in collagen, and then it really kind of takes off as you go from fibrosis stage 3 to 4. And this relationship is something we'll come back to because there's more to the fibrosis staging than simply the amount of collagen in the liver. So one of the things that we've taken great pains to incorporate in NAFLDsym and that allows us to, to be able to capture these relationships and compare them well to clinical data is we've really went through a lot of effort to make sure that our outputs from the model match up and can be used to compare with common measurements of treatment efficacy that you see from the clinic. So you can see at this table 3 different categories. On the left, plasma biomarkers, where we have outputs from the model that represent plasma triglyceride, ALT, ProC3, several other really common biomarkers that you'll see in NAFLD-NASH studies. Histology measurements such as the different axes of the NAFLD activity score, the steatosis, ballooning, inflammation, as well as fibrosis stage like we just looked at. And then lastly, on the right-hand side, the model has the ability to output different imaging measurements, for instance, looking at liver fat percentage in comparison with an MRI study or liver stiffness in comparison with an elastography study. Then if we move on to sort of swing back to the issue of fibrosis staging and specifically the comparison with the actual clinical outputs that you see in the literature there. One thing that we wanted to make sure to capture is within the actual NASH literature, there's really a long-standing guidance that scores the fibrosis stage based on not just the sort of the amount of collagen deposition, but also the location of where that collagen is being accumulated. So you can see here from this paper from Brunt, 1999, that they really the different zones of the liver are basically used to be able to stage the fibrosis. So in stage 1, you have sort of extensive accumulation in the paracellular area. In stage 2, that starts to move over and accumulate in addition in the periportal area. And in stage 3 or fibrosis stage 3, then you sort of fill in, so they refer to it as bridging, where you start to get then the accumulation or the deposition in the midlobular. And so what you'll see in our model, which is illustrated here in the bottom right, is that we've essentially replicated this submodel across the 3 different zones so that we can capture the spatial aspect that's used in the histology scoring, so that we can then line up, from a mechanistic perspective with how they actually do the evaluation in the histology studies. So one interesting aspect, though, that this brings up, and that we've noticed through the process of both building the model as well as running several simulation studies, is that you can get interesting disconnects between the actual efficacy of the drug, the resultant collagen loss, and the fact that you're looking oftentimes at a key efficacy measure as being the fibrosis score, so which of course is a discrete thing. So in this example, we have a hypothetical treatment in F3, F4 SIM cohorts, which are small subsets of the SimPops, where we've picked out 2 representative patients who, in response to the treatment, had basically equivalent loss of collagen in the liver, on an absolute basis. So you can see here, both the F3 and F4 patient had about 2.5 mg per gram liver loss. However, because those patients started in very different places with respect to their initial collagen level in the liver, if you looked from the perspective of the fibrosis stage, the F4 patient would basically look as if it hadn't had any efficacy from the treatment, whereas the F3 patient actually drops 2 fibrosis stages from 3 to 1. So this is sort of an important nuance and sort of an important learning that the model really helped us understand a lot better in terms of looking for whether a treatment's efficacious or not, and the possibility that your choice of clinical outputs may affect your ability to capture and to see whether the treatment was efficacious. So, sort of moving back to the issue of weight loss, we mentioned earlier in terms of validating the SimPops that one of the key things we wanted to look at in addition to these clinical relationships is the response to treatment or the response to the current standard of care. This simulation study that compares basically simulating weight loss over 1 year, such that at the end of the year, the patients had approximately 5% weight loss. We did that study and then we compared the simulation results to data from Villar-Gomez 2015 as well as Hamid 2018. And basically what you can see is in these studies, they had patients that lost approximately that much of their body weight over 1 year. They were NASH patients, and so they had access to the NASH score and its different components. And so you can see in the graph there that our simulation results line up really nicely. And actually, this is not calibration. This is actually sort of emergent behavior from the simulation, which predicted correctly these changes in NASH score in response to dietary intervention. So this sort of provides a validation of those SimPops. And then, in the interest of time, I won't go through this slide in detail, but the main point of this slide is to show that there's, in this case, Wong 2010, this was a nice study that looked at NASH patients, longitudinally across 3 years, and really there's a lot of nice data that correlates weight gain with NASH disease progression. So this is nice data, sort of outcomes data that is very consistent with our understanding of the mechanistic drivers of the disease, or some of the primary mechanism of driving the disease. And so with these things in mind, we want to end at least this little section on NAFLDsym with one example where we took what we understood about weight loss and weight loss as a standard of care and applied that to performing simulations with liraglutide, with a GLP-1 receptor agonist. So many of you are probably familiar with GLP-1 agonists. It's an analog, or they're analogs of endogenous active GLP-1. They're primarily used for the treatment of diabetes, type 2 diabetes. They're really effective at reducing glycemia, and they also induce weight loss in many patients. So in this simulation study, we're really focused on the weight loss aspect of the effect from liraglutide. So basically what we did is we took the study, looked at the study from a meta-analysis from Wilding 2016, where they looked at the exposure response from the liraglutide versus, compared to the body weight change from baseline. And then we basically created an exposure representation for liraglutide within NAFLDsym, and then capture these quantitative and dynamic aspects of weight loss in the NAFLDsym representation. And you can see in the bottom right here that we've captured quite nicely this exposure response relationship between the increasing dose of liraglutide with respect to how much the body weight was changed. So given the ability to do that, we're interested to see, okay, if we spun that forward, how then would liraglutide actually affect different aspects of NAFLD-NASH in NASH patients? And so now we ran basically a simulated clinical trial protocol for liraglutide for 1 year. Again, this is only the effects of weight loss of liraglutide, which have been implemented in these simulations. But you can see at the end of that year of treatment that you have a nice dose-dependent resolution or decrease in liver fat burden. And then if we look at the NASH score, then similarly we have a nice dose-dependent resolution or improvement in the NASH score as well, going from the placebo all the way through 3 milligrams. And then ultimately we're interested in fibrosis, and so we also looked at how the different patients improved in terms of their fibrosis score. And so you can see again dose-dependent improvement leading up to at the top dose of 3 milligrams. Almost 20% of the patients had an improvement in their fibrosis score of at least one fibrosis stage. So again, this was sort of a nice example to show how some understanding of the drivers of pathophysiology, as well as the exposure response in the PD, can allow us to leverage this model and get quite a bit more information out of it, predicting the effect on NAFLD NASH.
So now we, thank you, Grant. Now we bring back Arlene to introduce our next poll question.
Yes, thank you. All right, is your organization interested in understanding NAFLD, pathophysiology, and potential efficacy of novel treatments for NAFLD? Please make your answer selection. And let me remind you, if you have any questions for Grant or for Scott so far, go ahead and pop those into your questions panel. After Diane's example on IPF-Sym, we'll go into the Q&A session. I'm going to close the poll now. Okay, looks like we've got some good stuff. Diane, we're ready for you.
Great, thanks, Arlene. Yeah, so we'll dive right into IPF-Sym next. So idiopathic pulmonary fibrosis, or IPF, is a disease that primarily affects elderly patients. It has extremely high mortality rates. Following diagnosis, median survival time is just 2 to 3 years. And there can actually be substantial manifestation of pathophysiology before lung function is reduced enough to motivate diagnosis. The picture here shows that the architecture of the lungs in IPF patients can be drastically changed. And IPF is a progressive disease, meaning that lung function declines over time. One of the standard measures of lung function is FVC, or the forced vital capacity. And so we've recently developed a quantitative systems pharmacology model of IPF called IPF-Sym. And with IPF-Sym, we have the ability to predict effects of treatments on fibrosis, inflammation, and alveolar epithelial cells. As you've seen for our other platforms, we include pathophysiologically diverse patients, which we call SimPops. We're currently preparing to release the first version of IPF-Sym, V1A. And as Grant mentioned, for both NAFLDsym and IPF-Sym, we can use these platforms to support the development of new therapies by combining drug PK, PDGF, and pathophysiology to predict efficacy of new treatments. Again, we have flexibility in the framework to introduce new targets. And again, one of the important points that Grant pointed out is that we can evaluate combination therapies, which we'll see can be especially important for IPF, where the current standards of care have limited efficacy, and it may be very important to combine them with new therapies. So histologically, the lungs of IPF patients have 3 distinct zones. The normal parenchyma contains the unaffected healthy alveoli, and this region of the lung is the primary contributor to residual lung function. The honeycombed region of the lung contains collapsed non-functional alveoli, as well as an increase in extracellular matrix proteins. So this part of the lung can essentially be considered a wasteland, and it's not making any contribution to lung function. And then the third region of the lung, the fibroblastic foci, is where all of the active disease processes are occurring. So there's active remodeling of the lung that's happening as alveolar epithelial cells are dying off. They're producing mediators that activate fibroblasts, which then are synthesizing extracellular matrix proteins. Some of the mediators lead to activation of the immune system, which itself produces an array of mediators, which further support the fibrotic processes occurring in this region. So in IPF-Sym, we've included the 3 regions that I've just described. So again, the normal parenchyma has the normal healthy alveoli. Within this region, we have various cell types, including alveolar macrophages, both AEC1 and AEC2 cells. The AECs in this region, the ones that are adjacent to fibroblastic foci, can have increased vulnerability to apoptosis. And again, this region of the lung is making the primary contribution to lung function. And again, the fibroblastic foci region is where most of the action is happening. So we have AEC1 cells dying off. They produce a number of mediators that can help to recruit the immune cells. So things like neutrophils and macrophages become recruited and activated. The immune cells produce a whole array of mediators that some of them, such as TGF-beta and PDGF, can play a role in the activation and proliferation of fibroblasts. The fibroblasts are the cells that are synthesizing the ECM components, so things like elastin, collagen, proteoglycans, and some of the mediators actually modulate the dynamics of the ECM components. So things like MMPs and elastase can actually degrade some of the ECM components. And as the ECM levels increase, that leads to an increase in lung stiffness, which can actually feed back to further activate the fibroblasts in this region. And then the third zone, again, the honeycomb region, that's our kind of our wasteland. There's no functional alveoli, but an excess of extracellular matrix components. So no contribution to respiratory function, but the increase in ECM in this zone can also contribute to the increased stiffness that feeds back on the fibroblast activation. So then we can integrate the dynamics of each of these three zones, and they can help us to predict clinical outputs. So things like the FDC, high resolution computed tomography, the diffusing capacity of the lung, and 6-minute walk distance. And just one other point on this slide, you'll see some color coding carry through. So the alveolar epithelial cells are in gray, components of the fibrosis submodel are in green, and immune submodel shown in blue, and then clinical outputs shown in red. So as I mentioned, IPF patients there's a high mortality rate, but there can be a high level of interpatient variation in the rates of disease progression. So over a 1-year period, some patients can remain stable. This can be seen in the Blaster 2019 reference on the right, 2 different measurements, HRCT1 and HRCT2. There's a number of patients that have horizontal lines, meaning that imaging outputs such as extent of honeycombing are basically remaining stable. But you can also see a number of patients where the lines slope up, meaning that disease is progressing. And can also see this if we look at the forced vital capacity as a clinical output. So in Biondini 2018 on the left, there are fast progressors that have rapid declines in FVC, and compared to patients on the right that some remain relatively stable and others have a much more slow decline in FVC. So in IPF-Sym, we include both of those patient types, both fast and slow progressors. And similar to how variability was included in NAFLDsym, we basically take the same approach. Of course, this is specific to the lung and IPF here, where we're varying the parameters underlying the pathophysiology of IPF to generate a pool of potential patients, which we then validate with a whole number of clinical data sets. And further validation is performed by simulating the response to standard of care therapies, Nintedanib and Pirfenidone. So now we'll look at some of the outputs that were used to validate the SimPops. So starting with some of the outputs from the immune submodel. In our SimPops of 716 simulated patients, we'll first look at some of the immune cell numbers. So on the top, looking at alveolar macrophages, and have the simulated levels in our early and late progressors in the SimPops, and can see that that falls within the range that's observed from data from Blaster 2016 from lung biopsy data. And in the bottom plot, we have simulated and observed levels of alveolar neutrophils. So we can see that for this immune cell type, we're also seeing simulated numbers that are consistent with published data. So with the immune submodel, in addition to the cell types, one of the other important components are all of the mediators being produced by the immune cells. So actually just showing a subset of the mediators here, so things like TNF alpha, TGF beta, MMPs, VEGF, PDGF, and IL-1 beta. We can look at the simulated levels in our early and late progressing patients in the SimPops and compare those to clinical data from a variety of sources that's been compiled and plotted here as mean, minimum, and maximum, and can see for each of these mediators as well as ones that we're not showing that there's consistency between the levels in our simulated patients and that which has been observed in various clinical data sources. And then as far as some of the fibrosis model endpoints, again, looking at some of the ECM components. So total collagen is shown on the right for the SimPops versus data from Westergren-Thorsen, and lung proteoglycan levels are shown in the bottom versus the clinical data. So have good consistency as for our ECM components of the fibrosis submodel as well. And then starting to look at some of the clinical outputs. So first looking at some imaging outputs. So from images of the lung can get an estimate of the amount of lung that is in the fibroblastic foci region, as well as the extent of honeycombing. So you can see on the top, the fibroblastic foci are early and late progressors, have simulated extent of fibroblastic foci compared to 2 different clinical data sets. And on the bottom, the extent of honeycombing, also consistent with data, and you can see that there's a good degree of variation here. It can have just a very low percent of honeycombing up to 20 to 40% of the lung in this region. And then the extent of honeycombing fibroblastic foci also feed into our predictions for clinical outputs such as FVC and diffusing capacity. So we can see here simulated levels of FVC in our early and late progressors in the absence of treatment compared to data sets from untreated patients. And can see some between the early and late progressors, a little bit of a shift down in FVC. The same thing for the diffusing capacity. Some shifts down in from early to late progressors, and also consistent with the range of data from a number of different data sources. And then we can look at the FVC for fast versus slow progressors in our SimPops. So over a 1-year period, the fast progressors have a much more rapid decline in FVC, and this rate of decline in fast progressors in our SimPops is consistent with that rate seen in the BND 2018 patients, and our stable or slow progressors are more consistent with the slow progressors from that data set as well. So then, as I mentioned, there are only 2 approved therapies for IPF that have limited efficacy in mitigating the decline in lung function in these patients. So one of these drugs is Pirfenidone. It's a small molecule with anti-fibrotic and anti-inflammatory properties. In IPF patients, it's mitigated the decline in FVC. This can be seen in the King 2014 paper on the right, where there's a much more substantial drop in FVC in placebo-treated patients compared to Pirfenidone-treated patients. And preclinical data suggests that inhibition of fibroblast activation and procollagen synthesis may be mechanisms of action for this drug. And so we've used those mechanisms to represent Pirfenidone in IPF-Sym. And simulated the response to Pirfenidone in a Sim cohort of 30 patients from a subset of patients from our larger SimPops. And this subset was selected to have a group of patients that had a similar rate of progression compared to the placebo-treated patients in the King 2014 study. And then we took those patients and treated them in the simulations with 801 mg TID dosing for 1 year and can see that the pirfenidone-treated patients in our simulations have FVC declines that are consistent with those seen in the clinical data for this compound. And the second approved drug, Nintedanib, it's a small molecule receptor tyrosine kinase inhibitor. It targets PDGF, FGF, and VEGF receptors. The Recapitulates 2014 reference on the right shows that this drug also mitigates decline in FVC. And preclinical data suggests that inhibition of fibroblast proliferation may be the main mechanism of action for this drug. And so we use that mechanism to represent Nintedanib in IPF-Sim. And so for our simulations of Nintedanib, we first looked at the SimPops, the full SimPops in the absence of treatment actually had FVC declines that were consistent with that seen for the placebo patients in the Nintedanib study. So we used the full SimPops and treated those patients with Nintedanib for 1 year, and can see that the simulated FVC decline in our Nintedanib treated patients is consistent with that observed from the clinical study data for this compound. So the two standard of care therapies, Nintedanib and pirfenidone, were used to further validate our SimPops, and IPF-Sim is now well positioned to represent novel therapeutics with potentially different mechanisms of action. As mentioned before, we can look at combination treatment. So many of these drugs we may want to simulate in combination with the current standards of care. Could also look at combinations between two different novel therapeutics and assess potential efficacy. So I'll pause here to wrap up the IPF-Sim section. I think we'll have another poll question before we get to Q&A.
Yes, thanks, Diane. A final question for the audience. Is your organization interested in understanding IPF pathophysiology and potential efficacy of novel treatments for IPF? If you can take a few seconds to select one. Those that have not done so, you may send in your written questions for Diane and the rest of the group in the questions pane on your control panel. And we're going to go ahead and close the poll now. So it looks like we do have some interest, and with that, I invite Scott back as today's moderator of the Q&A. Take it away, Mr. Siler.
Great. Thanks again, Arlene. Thank you, Diane and Grant, for some great presentations. Before we get to some of the actual questions, I just available for licensing. NAFLDsym right now, IPF-Sim very soon. We're readying it for release as we speak, whilst also using it to evaluate various therapeutics, as Diane just illustrated. And not only is it available for licensing, but also services projects, you know, and actually it's not just or, it's an and. Sometimes we license and do the services project, train, and so everybody can hit the ground running when they're begin doing projects in-house. So let's see, I'll kind of leave it parked here on this slide when I pose, let's see, I think we have time for maybe 1 or 2 questions before we need to wrap up. Let's see, the first, this is for you, Grant. Aside from GLP-1, have you modeled any other NASH targets?
Yeah, so thank you for your question. Yeah, so we have you know, modeled over 20 different either targets or combinations of targets. Now, many of those were on the proprietary side, and so we can't divulge all of them that we've worked on, but there's many that are quite a few that we've presented publicly. So for instance, we've done work on ACC inhibitors, on FGF21, DGAT inhibitors, as well as AMP kinase. In addition to weight loss and the GLP-1, which we presented today. Thank you very much for your question.
Great, and there's another question here. This one's for you, Diane. Have you considered expanding into other areas of pulmonary fibrosis in addition to IPF?
Yeah, we're in a good position now where we've represented so much of the physiology of the lung that could definitely expand to other diseases in the lung. And as mentioned, some of our platforms like RADISym also impact the lung. So once we develop expertise in a certain organ, can definitely expand and continue to apply that in new areas.
So I'll just again conclude that here are some of our current offerings. You've seen that NAFLDsym and IPFsym are really nicely positioned to help support drug development in those areas. And I'll just again highlight what I mentioned in the beginning, that we are absolutely able and seeing a great deal of interest in developing custom QSP models for new diseases, perhaps a spin-off, if you will, say renal fibrosis or something completely new. Again, there are limited number of experienced and capable QSP modelers in the world, and we think we have a pretty good collection of them. So with that, again, I thank the audience for your participation, and that concludes today's webinar. Thanks, everyone.